We have a very well developed immune system. Our immune system has the ability to distinguish self from non-self molecule. So our immune system can say that these are the self molecules and these are the non-self molecules. These non-self molecules are normally, we call them, they are foreign, and these foreign non-self molecules, we can say that, other terminology we can use it, these are called the antigens. How our system identifies these non-self molecules? So therefore, if the non-self molecules are identified by using our lymphocytes. We have two kinds of lymphocytes. So, if the two kinds of lymphocytes, one is the B lymphocytes, other one is the T lymphocytes. They have a very efficient mechanism in order to differentiate self from a non-self molecule. An antigen can be defined or understood very well in terms of four terminologies. The first terminology is immunogenicity, and the second terminology is antigenicity. The third is allergogenicity, and the fourth is tolerogenicity. So these are the four terms which will clearly define what exactly an antigen is. What is the meaning of immunogenicity? Immunogenicity means the capacity to induce an immune response. So every antigen must have the capacity to induce an immune response. They are called the antigens. Antigenicity means the capacity to react with whatever is the product of immunogenicity. So, the capacity to react with the product of immunogen is what we call it antigenicity. So, these two terms are very, very important in order to understand the nature of or understand the detailing of antigens. In the third concept is allergogenicity. Allergogenicity is the capacity to produce different types of allergic responses. And the last one is tolerogenicity, which means the capacity for unresponsiveness. So these are the four terminologies that we must be familiar in order to understand the concept of antigen. Okay. Therefore, we can say that an antigen is that substance which will positively or negatively activates lymphocytes by combining with the B cell receptor as well as or the T cell receptors. That is an antigen. If it is a positive response, we will have the products that are being formed. If it is a negative interaction, so therefore there will be non-responsiveness or what we call it a tolerance. When we look at antigens, there are two properties of an antigen. So the two properties of an antigen is, one is called immunogenicity. This is one of the properties of antigen. What exactly mean by immunogenicity? When you allow an antigen to interact with the lymphocytes, so therefore let us say that antigen interacting with the B cells, what will be the response? You can have, when it is a positive response, you can have either B effector cells, okay, B effector cells and either B memory cells. So these are the two kinds of responses. Either B effector cells will result into, these are the plasma cells and that will give rise to antibodies. You see a product is being formed by interacting with the antigen. Suppose if this antigen interact with the T cell. That is another kind of lymphocytes. When it is interacting with the antigen, you will have a T effector cells and T memory cells. Okay? Examples of T effector cells are nothing but either T cytotoxic cells, cytotoxic lymphocytes, or the T helper cells. All these are examples of T effector cells. So this capacity to induce an immune response is what we call it the immunogenicity, which is one of the property of antigen. In the second property of antigen, this is the first property of antigen. The second property of antigen is, it is called antigenicity. What is the meaning of antigenicity? Antigenicity means it is the capacity to combine with the product of immunogenicity. That means what are the products? The products, one here, the product is an antibody. So an antigen must be capable of combining with the antibody. Then only it becomes a true antigen. And this property is what we call antigenicity. In other words, an antigen must have two properties. One is immunogenicity and the second one is antigenicity. Please understand that all immunogens are antigens. That means they are foreign. Whereas all antigens cannot be immunogenic, which means all foreign substances cannot induce an immune response. Look at, for example, a safety pin or the gold the ring that you wear. They are foreign, but it may not be inducing an immune response. So therefore, all immunogens are antigenic, whereas all foreign substances or antigenic substances may not be immunogenic. So this is one clarity that we need to have in the very beginning itself. If all antigens are not immunogenic, then what makes a foreign substance immunogenic? This brings us to a very important properties of antigens. There are four main important properties of antigens, and the first property of an antigen is, first of all, foreignness. So an antigen must be foreign. It means it has to be a non-self molecule. Higher the phylogenetic distance, better is the antigen. Look at certain antigens like, certain examples like you would have heard about BSA, that is the bovine serum albumin. You know that bovine serum albumin you inject into a cow. It is not antigenic, it is not immunogenic because it is taken from the same cow. But you inject that into, let's say, into sheep. It may not be a very good antigen because if the phylogenetic distance between a cow and a sheep will be less. But you inject that into a man, or inject that into a rabbit, or inject that into a chicken. It can be a very good immunogen because the phylogenetic distance is greater. At the same time, let us look at sperm or the corneal proteins. Even if you take it from the same individual, inject the same protein into a person, they are very good antigens. Why they are very good antigens? Because these sperm, corneal tissues, etc., they are not into either regular contact with the lymphocytes. Our lymphocytes will treat sperm protein as well as the corneal protein as foreign, and immune responses can be induced. Look at the cytochrome C. Cytochrome C is a protein which is, you will see across all the species, starting from bacteria to the highly evolved organism. And this protein, since there is a common origin, this protein cannot be used as an antigen. because of the common originality. Look at another classic example is collagen. So collagen in all the species, the variation in collagen is very minimum. Because there is a less variation, you will see that collagen cannot be a very good antigen. So I'm giving the characteristics of what makes an antigen a strong immunogen depending on the phylogenetic tree. Another very good example is, if you have heard about the MHC molecules, the MHC molecules are very good antigens. That is because within species you will find a lot of variation in species. So MHC will act as an excellent antigen. That tells us that there can be two kinds of antigens. One is what we call an alloantigen, that is within the species is an alloantigen. Other one is a xenoantigen. Xenoantigen means it is outside the species. So these are the, depending on the foreignness, depending on the distances, we can say that these are the two kinds of antigen that are, we can group them into these two categories. This is the first property of antigen. The second property of antigen is what we call it the molecular weight. Molecular weight. Generally, any molecule with greater than 10,000 Dalton molecular weight is considered to be a good antigen, and up to 1 lakh Dalton molecular weight is considered to be an excellent antigen. Anything with a molecular weight less than 10,000 is a very poor antigen. We will look at such molecules later on in the context of haptens. We will be looking at them. So this is another characteristics of another property of antigens. Now with that, when we say that the molecular weight is important, we should also understand that either monomeric units of proteins, which are amino acids, or the monomeric units of polysaccharides like monosaccharides, or nucleotides or fatty acids, they may not be acting as a very good antigen. Only the polymeric form can, with a very high molecular weight, are capable of acting as a very good antigen. Third property of a very good immunogen after foreignness and molecular weight is the chemical complexity. Chemical complexity. So in other words, an antigen or an immunogen must be chemically complex. As we have seen, monomeric units are not chemically complex. Inorganic substances are not chemically complex, so they cannot induce immune responses. Look at Teflon, look at polyacrylamide. All these substances cannot induce an immune response just because they are not chemically complex. Look at the gold that you wear. It is an addition of gold particles, AU. It is not chemically complex. That is the reason why it cannot induce an immune response. In comparison, you look at the complex molecules like proteins. Proteins, higher the complexity, greater is the immunogenicity. For example, in the primary structure, the secondary structure, the tertiary structure, the quaternary structure, if the protein is in native form, greater will be the complexity and greater will be the immunogenicity of that protein. Certain very specific amino acids like aromatic amino acid and very specifically tyrosine is highly immunogenic. You see how complexity and certain characteristics makes a molecule highly immunogenic. So this is, the complexity is another important property of immunogen. And the fourth important property of immunogen is biodegradability. An antigen or an immunogen must be degraded. degraded by the pathways that we have within our system, like the endocytic pathway, etc. It should be able to be degraded and should be able to present with the help of antigen presenting cells to the lymphocytes. Then only immunogenic responses can be generated. So the antigen has to be biodegradable and should be able to be processed and presented by using antigen presenting cells to the lymphocytes. We have looked at the most important properties of immunogen. One is foreignness, the high molecular weight, chemical complexity, and as well as the biodegradability. There are other characteristics as well. But looking at all these, we understand that proteins suits the best. Proteins are the best antigens available. You will see many of these are present in protein. Characteristics are present in proteins. Lipids, by and large, they are not very good antigens. However, you can club lipid with the protein and you can make them a very good antigen. For example, you would have heard about the test, Wassermann's test for syphilis. So, the antigen which is used is cardiolipin. Cardiolipin is, as you know, it is one of the phospholipids. Now, this cardiolipin combination with the protein can act as a very good antigen. Nucleic acids, they are not good antigens at all. However, antibodies can be produced against the nucleic acid with the help of nucleoproteins. You would have seen that antibodies are raised against nucleic acid in certain systemic lupus erythematosus. Antibodies are raised against nucleic acids. So all these are possible in combinations with the proteins. Otherwise, by and large, proteins are the best antigens. Then can be carbohydrate, lipids and nucleic acids are not at all used as good antigen. They cannot induce immune response. We have seen properties of a good antigen or a good immunogen. But are these the only properties which will differentiate a very good immunogen from a not so good immunogen? There are many more other parameters. One such parameter is the genotype. In the genotype of the animal which is being immunized. Please understand that the MHC molecule, that is the major histocompatibility complex molecule, controls immune responses. We have seen that it can vary from species to species, individual to individual. There can be variation in MHC. So that this determines the immune response. And that is regulated by two things. Number one, is the antigen being processed properly and presented with the help of MHC. So how is the processing has occurred? Now the second aspect is, has the lymphocyte got the receptors? Lymphocytes, has it got the right receptor to identify the processed antigen? These two factors will determine if the response an individual, an immunized animal makes towards a particular antigen. So therefore, genotype of an organism is very, very important. Another important aspect is the dose that an antigen is given, the dose. If too much of antigen is given, there can be a possibility non-responsiveness. Too much is given, it suppresses the immune response. If too little is given, same non-responsiveness, because there is not enough dose to induce an immune response. Therefore, if the dose of an antigen is very, very crucial. And the third aspect of inducing an immune response is the route of its administration. So, in the route of administration, there are several ways by which an antigen can be administered. If the standard word is parenterally. Parenterally means routes other than orally. So which are the routes other than orally we can administer an antigen? Different ways. One possibility is, you know, that it is intramuscular. Through the muscles we can insert an antigen. You can have intradermal. We can have intradermal inside the skin. We can have it in subcutaneous. Beneath the skin you can have it subcutaneous. You can also have it intravenous. Intravenous, inside the vein we can introduce an antigen. We can also have in the peritoneal cavity. That is in the peritoneal cavity. If you recollect, in earlier times the rabies vaccines were introduced in the peritoneal cavity, and we can also give vaccines orally, like polio vaccine. Now the polio vaccine is given orally. Earlier it was given in, like intradermal. So these are the ways by which an antigen can be inserted, and it is very important that an antigen must enter through the right route. Another important aspect about immunogenicity is the charge of the molecule. So though the charge is normally, it is immaterial charge of the antigen. It is normally immaterial. However, if it has got too high a charge, it will suppress an immune response. So it should not have too much of a charge on the surface of the antigen molecule. So this is another important aspect. And the last aspect is it's about the conformation. This comes especially in the case of protein molecules. It is observed that a native protein is more antigenic or more immunogenic than a denatured protein. What is likely to happen? As we proceed, you will understand that when a protein is denatured, in the site which is inducing an immune response, these are called the antigenic determinants, that may be getting disassembled. So therefore, if the native form of a protein is more immunogenic than the denatured form. We have seen many aspects which will make a protein or an antigen highly immunogenic. We have seen that it is a chemical complexity is important, foreignness is important, molecular weight is important, it should be biodegradable, and then we have also seen several aspects like genetic distance, in the dose that is given, in the route of its administration, and finally the charge of the molecule, and also the conformation of the antigen which is inserted. These are certain aspects, certain characteristics of antigen.